Hi, my name's Andrew Murray. I work at Harvard University, and I want to give you part 2 of how to shmoo and find a mate. You'll find out what a shmoo is in a second. If you want to go back to things more basic, there's an earlier version of this talk, but everything that was explained there will at least get touched on here, so you can probably watch this without the first talk if you're feeling adventurous. So here we go. So the first thing, and actually one of the most important things to do in science, is to introduce the heroes. So this work is an example of what is now often called systems biology, and that means bringing together different parts of biology with ideas and tools that come from the mathematical and physical sciences. So this person is Mathieu Peel. He was a French postdoctoral fellow who worked in my lab. He is a very fantastic cell biologist, and this is Qinling Guo, who has a very interesting history. He was born in Taiwan. He went to medical school, then he got a master's degree in electrical engineering, and then he did what he'd always wanted to do, which is to do a PhD in theoretical physics, and he came to work with us after that. And so Mathieu is more experiment, and Qinling is more theory, but together within, the three of us had a very synergistic interaction where all sorts of things happened where, in retrospect, no one can figure out quite exactly who did what to make things work. So the other hero of our story is our friend the budding yeast, and we work on yeast like lots of people do because yeast are simple. If you want to understand basic principles about how biological systems work, the best way to do that is to find the simplest possible example that you can instead of the most complicated. And yeast are simple and we're complicated, which is why we work on yeast. And so here's the steel cage match. That's us, the peeps. This is our Latin name, Homo sapiens. Here are the yeasts, and this species is called Saccharomyces cerevisiae. This is the baker's, brewer's yeast. This is what makes beer and wine and bread, which many of us depend quite heavily on. And here are some of the differences between them, all of which, at least on this slide, are in yeast's favor. Yeast can't do science, which is why we're around. But if you look at us, it takes 20 years to make a new person. By the time you're born, grow up, mate, produce progeny, it takes a yeast cell 90 minutes, an hour and a half. The other thing is during those 20 years, you start with a single cell that was made by the meeting, the propitious meeting of your dad's sperm and your mom's egg, and that cell divided and divided and divided until we get you, which is 50 trillion cells, big complicated multi-part machine. The organism in yeast is one cell. When that cell divides, that's 2 new yeasts. And when it divides again, it's 4, 8, 16, and so on. We have in each one of our cells, each one of yours, each one of mine, about 6 billion bases of DNA. In a yeast cell, there's 500 times less. It's about 12 million bases. And so everything's simpler. There are fewer genes. But the biggest difference of all is there aren't so many experiments you're allowed to do on your friends and families, and we're certainly not going to get to do any forced mating with people. Okay? It's against the law and all sorts of moral standards. In yeast, anything goes. And anything includes genetic manipulation of incredible sophistication and variety. It includes forced mating, so no one tells us we can't force cells to mate with each other. And there's the sort of extreme version of survival. We do things where everyone except a very few special cells that we want to study die. Even Fox isn't allowed to do that. Okay? And at the bottom of the list, there's something that everyone does, at least everyone who's a eukaryote, they have sex. And so if we're interested in what some of the fundamental features are, how cells find each other, how they pick a partner, we can use this incredibly simple organism to study that and try and draw lessons that we can apply to all sorts of other parts of biology. So I'm going to walk you through the life cycle of budding yeast. So here is a cell as it's born. So the red thing in the middle is its nucleus. It's going to grow. It's going to pooch out this little bud here. The bud will get bigger and bigger over time. And eventually the cell will divide to make 2 cells, a smaller cell and a bigger cell. And you can just keep going around that circle indefinitely. This is a haploid cell. There's 16 chromosomes, 1 of every sort. And it's called an A cell because yeast comes in 2 sexes or mating types called A and alpha. Here's an alpha cell. Everything looks exactly the same because everything is exactly the same except for one pair of genes which determines which way you mate. So these cells do the same thing. They make a bud, they grow bigger, they divide, and around the circle again. And the way they talk to each other is they make and respond to pheromones or perfumes that each other produce. So the alpha cell makes something called alpha factor, which can diffuse, bump into an A cell, be detected by a receptor on the surface of that A cell. So the one on the A cell is called the alpha factor receptor because it's detecting the pheromone that comes from the alpha cell to the A cells, and vice versa. A cells make a pheromone. It can travel through water, bump into a receptor on the surface of the alpha cell. So this one is called the A factor receptor because it's receiving molecules that are coming in this direction here. And when cells detect those pheromones, instead of budding, they stop dividing, they grow towards each other. So they make these little protrusions that are called shmoos after a cartoon character from the 40s and 50s. And when those shmoos grow towards each other and touch, the cells fuse, the nuclei fuse, so now instead of being 16 chromosomes, they're 32, 16 from each parent. And this cell in turn can go through a cycle of growth and division. So it's going to divide 2 cells, this cell will bud, and so on. If you starve these cells, they can go through the specialized division of meiosis, which you can see down here. That makes 2 A and 2 alpha spores. You've gone from being a diploid to a haploid. So if you like, this is the yeast equivalent of eggs and sperm. And if you treat them with the right conditions, they germinate, and you go back to haploid cells that can go round and round and round the cell cycle. And so there's a lot of genetic control we have over this in the laboratory, so we can decide whether we want cells to grow as haploids. We can set up matings where we can quantify how many A cells mate with how many alpha cells. We can grow diploids. We can put them through meiosis as part of the genetic manipulation that produces the strains that we need for our experiments. So what I'm going to do now is walk you through a molecular view of sexual signaling. And what I need to do before I do that is to explain for 2 things. First of all, it's going to be a sort of complicated slide, so I'll try and go slowly. And the second thing is the details on this slide aren't desperately important for understanding the rest of the talk. It's to explain to you that we know a lot about what's going on, and because we have genetic tricks, we can manipulate various parts of this process. There's one part that will get manipulated in the talk, and I will tell you exactly what that part is when we come to it. Okay? So we're going to start at the beginning. The crucial thing is there's outside of the cell and inside of the cell, and those 2 things are separated. If you look here, my hand's going to go back and forth along this gray, light gray line with 2 darker gray lines on the outside. That's what's called the plasma membrane. That's what separates the inside from the outside. There are proteins that snake back and forth through the membrane. This one here is the alpha factor receptor. And the alpha factor that's going to bind to it is this little black horseshoe over here. When that binding happens, this protein here, which is made up of 3 separate pieces called alpha, beta, and gamma, and is called a G protein, is going to interact with the receptor, because what's going to happen is the pheromone binds to the receptor changes its shape or its conformation, to use the technical word. That induces this G protein to bind to it, and something interesting is going to happen. This G protein is called a G protein because it has guanine nucleotide diphosphate bound to it. So 2 phosphate groups are on this molecule. When this molecule cozies up to the receptor, it induces a change in the shape of the G protein, and that causes a swap of guanine nucleotide triphosphate, so 3 phosphates, and that in turn changes the conformation of the alpha subunit. You can imagine it getting all bumpy, and that pushes away the beta and gamma subunits. Okay? And these are what's going to do the business of signaling, and the way they're going to do that is they're going to recruit what's called a scaffolding molecule. So this molecule in this light blue color is called FAR1. This was isolated by geneticists, so FAR stands for pheromone arrest. It recruits another protein. This protein is called CDC42. CDC stands for cell division cycle, so this is important in the whole process of helping cells grow and divide. The names here are really not very important. I'm just telling you how people found them. This protein does 2 things. It stimulates the polymerization of these little balls to make long rods. These are what are called actin filaments. They are amongst the railroad tracks of the cells. And so molecules that are going to arrive and be incorporated at the membrane have to travel down these up to the plasma membrane. And they'll be important, very important for our story. The other thing that CDC42 does is to activate this class of molecules that are colored a light red, which are called protein kinases. These are molecules that modify the chemical structure and therefore the activity of other proteins. Sterile here means that these genes were isolated by geneticists who found mutations that made sterile, that is, they could not mate with each other. So sterile 20 is a protein kinase that modifies the structure and activity of sterile 11, which in turn modifies the structure and activity of sterile 7, which in turn modifies the structure and activity of Fuz3, and ultimately this leads to the transcription of genes that are required for cells to mate with each other, and it leads to the cell cycle arrest so that the two cells are both in the same part of the cell cycle when they mate with each other. Okay? So this is how signaling works. There are three more things that I have to talk about in this slide. And the first is about what a biologist or a physicist would call positive feedback. That's a scheme where the rich get richer and the poor get poorer. And if we want our cell to stop growing uniformly and start growing asymmetrically, which is what it's going to have to do if it's going to make a shmoo, we need some sort of differentiation between different parts of the cell surface. So someone has to get richer and someone has to get poorer. And so what's going to happen here is CDC42 is going to stimulate actin polymerization right here. But these actin filaments, I told you before, are the railroad tracks of the cells. They're the things along which molecules that end up in the plasma membrane, like the alpha factor receptor right here, get delivered. So if you have more polymer, you're going to deliver more receptor. But if you have more receptor, you're going to do more signaling, you're going to activate more CDC42, you're going to have more actin, you're going to deliver more receptor, you're going to have more actin, you're going to deliver more receptor, and so on. So the rich will get richer, the poor will get poorer. Some part of the cell will suddenly start to grow asymmetrically. Okay? So that's turning things on. We also need to be able to turn things off. Once the pheromone's gone, the cell has to stop signaling because we don't want to try and mate anymore. We want to grow and divide. And so one of the ways that happens is shown here. This is a protein called SST2, and SST stands for supersensitive to pheromone. And this is involved in stimulating the hydrolysis of this GTP back to GDP, which gets rid of all these little bumps here. The alpha subunit can bind the beta and gamma subunits, the scaffolds fall off, the signaling stops, and we're back at gram zero. There's another way of recovering. There's a way of recovering even if there's pheromone still present. And that's because if you're an A cell, you secrete this molecule here. So this little Ms. Pac-Man here is what's called a protease, a protein that cuts other proteins up. And this protein snips alpha factor. And after the snip, the alpha factor can't signal anymore. This is a crucial protein, as you'll hear later in this talk, to help explain why yeast cells can mate under a very wide range of conditions. Why if you just take two cells and cozy them up to each other, like a man and a woman on a desert island, they will mate successfully. And why at the other end of the spectrum, you can recreate Studio 54 in New York with thousands of sweating, heaving people and have almost everyone find a partner and go home satisfied at the end of the night. And you'll hear that this protein is crucial to explaining how that happens. And so in the talk, there are going to be two halves. In the first half, we destroy this protein. Because if this protein isn't destroyed, we never know quite how much alpha factor is there because Ms. Pac-Man is busy chewing it up all the time we're doing the experiment. So the first part of the talk gets rid of this complication because genetically we destroy the gene for this molecule. The second half of the talk, where we're interested in understanding how cells mate successfully under all these different conditions, we bring it back. I'll try as hard as I can to make the distinction between the first half and the second half as clear as I can. Okay. So this is what it looks like. This is a movie is worth 1,000 pictures. Here are two cells, one here and one here that are going to mate with each other. And what's going to happen, they will grow towards each other, they'll touch about here, and they'll produce their first daughter over here. Okay? So here we go. Growth, touch, fusion, daughter. Okay, and I'll go back and do that one more time. Here we go, growing, meeting about here, fusing, and there comes the daughter right there. Okay? This whole process has been speeded up. In reality, that whole movie takes about 2 hours. So we're seeing something enormously speeded up for the screen. But we can watch it if we take slow enough pictures with a microscope. Okay? So what I want to talk about now is the first part of mating, which is this business of shmooing and finding a source of pheromones. So here we have the concentration of pheromone indicated in this reddish color. This is low and this is high. This is a cartoon of 5 cells stuck in a gradient of pheromone like that. And what will happen if we watch cells, and we'll show you data from an experiment like that later in the talk, is every one of these cells will shmoo, and their shmoo will grow up the gradient towards higher and higher concentrations of the pheromone. That's called chemotropism. Yeast cells can't swim, they can't walk, they can crawl, they can only grow. So chemotropism means growing towards some source of attractant. But what happens if instead of making a gradient, we take another cartoon 5 cells and we put them in a uniform concentration of pheromones? So the pheromone concentration is the same everywhere. What happens is these cells don't just keep getting bigger and bigger. All of a sudden, they too make little shmoo tips, but they do it in random directions. And this phenomenon of symmetry breaking happens again and again in biology. In fact, when your mother's egg was fused with your father's sperm, that cell divides, and at that stage we don't know which is front and back, which is head and tail, but later on during the development of the human embryo, something happens to break the symmetry and we get a top and a bottom, a front and a back. And this is an example of that process, and a process which we can study in a much simpler and easier to manipulate experimental system. Okay? So I need to tell you what some of the things are that we're going to look at as we follow cells in this talk. So here is a yeast cell. The blue thing is its nucleus, where the DNA is. We're going to look at 3 different sorts of components. The way we look at them is we fuse the proteins to a fluorescent protein originally derived from a jellyfish. And we can see those proteins underneath the microscope. And because they're different colored fluorescent proteins, we can look at multiple different proteins in the same cell. And we can watch those cells do things in real time. So here are the things we're going to label. This red color here is a protein called SPA2, which forms part of a structure called a polarisome. It's the thing that tells the cell where to start nucleating actin filaments, the things along which everyone's going to run. And those actin filaments, or cables, which is a bunch of filaments stitched together, are shown in this purple color here. We don't mark the actin itself because it's a sensitive protein. It has to interact with lots of different things. We mark a protein called actin binding protein 140, which binds to the actin cables, but it still tells us where the actin is inside the cell. And the green ring here is a class of proteins called septins, which are important for controlling cell polarity and also controlling the rigidity and integrity of the cell wall. So this cell, if it never saw pheromone, is a cell that's about to bud. It's growing uniformly, but if we can see the polarisome at this side of the cell, we know for sure that the cell will bud in this direction. So if you imagine the cell just tilted up this way here, here's that cell having made a bud. And when that cell comes to divide, those same proteins, the septins and the proteins that make up the polarisome, are here where the cell has to pinch in half to produce 2 daughter cells. If instead we treated this cell with pheromone, so that's what alpha factor stands for, that's the pheromone from alpha cells, the cell will shmoo instead of budding. It's still true that the SPA2 and the polarisome will be the tip of that growing shmoo. There'll be a gradient of septin molecules behind it, making sure that the cell wall doesn't pooch out the way it would if you were going to make a bud. And there will be the actin cables running back, because they're the things, as I talked about in the previous slide, that are going to transport everything towards the tip of the cell. So now what we want is we want different models that are going to try and explain how it is that cells can break symmetry even if there is no gradient in the pheromone. How is it that if the signal is the same everywhere you look, you can still end up deciding to make a shmoo? So here's the first model. This model says that here is this structure, the polarisome, that tells the cell where it's going to bud. So why not just use this structure, which already exists, and use that instead when the cell sees pheromone to tell the cell where to shmoo? Okay? The second model is derived from work of the great British mathematician Alan Turing, and it's something that's called lateral inhibition, or a reaction diffusion scheme. So in this cartoon, we imagine there is some stimulus that turns on some activated molecule inside the cell, which we just call A here. A can activate more of itself, so there's some sort of positive feedback loop where if you activate a little bit of A, it gets easier to activate more. This positive feedback loop works over a short scale of distances, but it is also true that the activator turns on I, which is an inhibitor. So that's why this arrow is colored red and has a blunt end, because I is going to turn off A. This works over a longer range, and so what's going to happen with this is close to the source of the stimulus, you're going to activate things more. Further away, you're going to inhibit them. And so you can use schemes like this to explain all sorts of patterns. For example, the patterns on seashells of black and white. This class of model predicts that if you take a cell, let's say it's a spherical cell, like a yeast cell is almost spherical, and you give it weak stimulation, you'll see a little zone of activation, the green here, and then behind it, there'll be this zone of inhibition. But the math that goes behind these models predicts that the more you stimulate the cells, the harder time the inhibition has, and the more likely you are to produce 1 and 2 and maybe 3 and maybe 4 different zones of activation. So there's one here, there's one there, and there's one there. And therefore, that cell would be trying to polarize in 3 different directions at once, which, since you're only allowed to mate with one cell, is not going to work very well. There's no yeast bigamy. Okay? And in particular, this model also predicts that if transiently, briefly, you see 2 zones of activation, because the concentration of the inhibitor is highest here right between them, they're going to push each other apart. And so you'll either end up with 2 on opposite sides of the cell, or one will win and the other will lose, so you end up with only one. Okay, so that's our second model. Our third model is something that Chin Lin, as far as we can tell, came up with that is novel, and it's called global integration. And it relies on 3 things. It relies on some set of railroad tracks, what a biologist would call a cytoskeletal polymer, like actin. And it relies on things being transported along them. So when they get to the end and they get to the membrane, they end up being red like this. These are the active signaling molecules. If you like, this is the pheromone receptor. They have an inactive form, which is here yellow, and there's one right here in the center of this cell. And it's going to get transported in or on vesicles to the surface of the cells, and it can't be active until it's at the surface of the cell. If it was the receptor, it can't be active till it's at the surface of the cell, because it can only detect the pheromone, which is outside the cell. And so the model has a very specific requirement, and that is that when these things get transported, the transport is what a biochemist would call distributive. And what that means in regular words is you bind to a filament, you move along it, you fall off, you bind, you move, you fall, you bind. Okay? What that means is that a vesicle like this is going to take a random walk. It first goes in one direction, and then, for example, goes this direction. Eventually, wandering around, it bumps into some part of the surface of the cell. And at least in a cell like this, where all the filaments are evenly distributed over the surface of the cell, the mathematical operation that this vesicle is doing, which is adding up all the different vectors which describe the directions of transport, ends up being 0. There's no net force pulling this in one direction rather than another. But if it gets delivered to a particular point on the cell, which it will eventually, like this one here, now there's more signaling on this side of the cell. And if you add up the transport directions associated with each of these green filaments, there's a small arrow that points in this direction. So now a new vesicle that hits the filaments is more likely to end up on this side of the cell than it is on this side of the cell. Okay? And if you keep going, the rich will get richer. Okay? Because the more stuff you deliver over here, the more filaments there are pointed towards this side of the cell, the bigger this arrow gets. And so this sort of scheme can explain how the cell polarizes. Like every model, it has good things and bad things. The good thing is, without any sort of fancy fiddling with the math, there's a very robust drive of these cells to polarize in one and only one direction. The con is that that drive to polarize is so strong that it can overcome the asymmetry that's imposed on the cell by a gradient of pheromone. So you can imagine that one way of getting more molecules on this side of the cell than the other is there's a little more pheromone over here than there is over there, right? That will help this process. But if the drive is so strong that little random fluctuations can tip the balance quickly, the cell has a race between detecting the gradient in pheromone between one side and the other, and one of these random events running away and polarizing the cell. Okay? So now we're going to compare the three models, and we're going to do it by looking at a graph. So on the x-axis is the efficiency with which you transport things towards the surface of the cell. On the y-axis is how hard we're stimulating the cell, how much pheromone we're adding. Here's the historical mark model, the first one where we're just going to use the instructions that tell you where to bud to tell you where to shmoo. It's pretty simple. Above some threshold, there's no polarization. Above it, the cell's polarized. We know where they're going to polarize. They're going to polarize where the mark was. Here's lateral inhibition. If we don't stimulate or we don't have enough transport, we don't polarize. We weakly stimulate, we get one zone of activation. We strongly stimulate, we get 2, we get 3, we get 4. Okay? And global integration is exactly the opposite. If we stimulate strongly, we only get 1. If you stimulate weakly, we get 1. Sometimes we get 2 as an intermediate before we get 1. And if stimulation and transport are too weak, we don't get any. Okay? So this is what things look like after the system settled down. That's what a physicist would call, or a biologist, achieving steady state. We can also ask about kinetics. We can ask about how fast things get there. The historical mark is sort of boring. Here's the little historical mark. Everyone gets sucked there, and the cell ends up polarizing in this direction. With lateral inhibition, you can transiently get 2 zones of activation, 2 places where you've accumulated signaling molecules. But as I told you before, they're going to push each other apart. So either they're going to end up on opposite sides of the cell like this, or one is going to win and one's going to lose, and you end up with just 1 zone. Global integration makes exactly the opposite prediction. It says that if you have 2 zones of activation, and now we add up the transport direction, let's say we have 2 things that are pointing like this, the net direction of transport is going to be right between them. And that's going to pull the 2 zones of activation towards each other, which is what you see in this diagram. So they're going to move towards each other, and eventually they'll fuse. So what we want to do is test the predictions of these different models. And particularly these 2 models are useful because they actually make the opposite predictions for the same experiment. So someone more or less has to be right. Okay? So the first thing we wanted to do is we wanted to ask, is it true that cells use the information that was supposed to tell them where to bud to tell them where to shmoo? Okay? And so the way we do that is we look at a cell like this, which is just divided. In haploid cells, the rule is that you make your next bud right next to where you made your last one. So we know where these cells are going to bud, right where these red dots are. We add alpha factor. We wait until the cells either bud or shmoo, we don't care which, and we measure the angle between where they were expected to bud or shmoo and the direction they actually polarized in. We call that angle theta. We convert that into the cosine of theta, which varies between +1 and -1. We average that for lots of cells treated with the same concentration of pheromone, and we draw a graph. Here is the mean value of cosine theta, a measure of the average direction of polarization, and here on a logarithmic scale is the concentration of pheromone, going from 0.1 nanomolar up to 1,000. So there's a useful thing to know, which is our experiment suggests that no cell in the wild-type world ever experiences a concentration of pheromone much greater than 1 nanomolar. Okay? So the first thing to see is down here where the pheromone concentration is very low, cells bud in the same direction that they would bud if there was no pheromone, which is what this green dashed line is here. And up here, they're still all budding. Down here and above, they're all shmooing. There's a transition in between. And what you see is at the concentration of pheromone, which is the physiological concentration, the one that cells actually experience, cells are actually going slightly in the opposite direction of the direction we expected them to bud in. Okay? As the pheromone concentration goes up and up, you start to come back up here towards the direction that the cells were expected to bud in. And what we think is happening here is that these cells polarize so fast that they actually do use the information that was going to tell them where to bud. But mating never happens anywhere like this. Okay? So this experiment suggests, at least under the conditions where cells normally mate, they are not using historical marks to tell them which direction to go in. Okay? So here's the next experiment. So this is a slightly complicated experiment, so again I'll try and explain it clearly. We've marked two things inside these cells with different fluorescent proteins. This is CDC42, that protein that stimulates actin polymerization. And here we have CDC10, which is one of the septins, the thing that gets deposited sort of just behind the tip of the shmoo. And we show a picture of the cells in green, a picture of the cells in red, and down here we overlay the two pictures so we can compare the distributions with each other. Okay? These are times in hours and minutes, and they're counting backwards, if you like. This time 0 is the first time that the cell visibly started to polarize. That is, we could see under the microscope it's growing in one direction, and so we work backwards. That's 5 minutes before, 10, 15, an hour and 15, and 3 hours before. Okay? And the other thing we do, because there's so much green here, it gets sort of hard to see stuff. For all these time points, we subtract this initial level of green, and so we just see where the green concentration has gone up relative to that initial point. Okay? And what you can see is, here the cells are pointing in one direction. Here they're pointing in two directions, and you can actually see there's more CDC42 here, and there's more here, and there's a little gap in between. Okay? So there are two little patches. What lateral inhibition says is those patches should be pushed apart. Global integration says they should be pulled together, and if you watch, what's happening to them is they get pulled together, and at our 0 time, the time in where the cell starts to make a shmoo, it started to shmoo out in this direction. And you can see, if you compare the overlays with the individual colors, that the CDC42 and the septin are in the same place. So it's all part of the same polarization machine. So the next piece of evidence is to ask what happens if we really overstimulate cells, so that if we just, put Viagra in the water and make all sorts of excitement happen, what happens to cells? So this is a concentration, 10 nanomolar alpha factor, that cells normally never see. Okay? And what we're going to do is we're going to show you a movie. There's a little green dot. That's the polarisome, SPA2, labeled with yellow fluorescent protein, although it's green in our movies. And what you're going to see is the cell, first of all, it will start to polarize down here. Then it will quit. You can imagine it quitting in frustration. And it will polarize over here. And it will do a couple of different directions that it tries to grow in, but it never is growing in two directions at the same time, and it's never showing two spots of SPA2 at the same time. So here we go. First, second, third, fourth, and the last one will be down by the first one again, down there. Okay? And so this is not doing, again, what lateral inhibition would have the cells do. Because lateral inhibition says if you stimulate the cells too much, you'll get two zones of polarization, and the cells will grow in two directions at once. I should point out that none of the models quite explain why the cell stops growing in one direction and picks another one, and that's an interesting subject for future research. Okay? So what I'm going to tell you is that so far, and the truth in science is always a work in progress, things change as people get more evidence. There's evidence against historical marks, there's evidence against lateral inhibition, and so the only person left standing at this point is global integration. That doesn't mean this is the right model, but at least it means it seems like it's more in the right direction than the other models. So one of the things that we claimed is one of the problems, if you like, with global integration as a model is it suggests that cells can only detect gradients of pheromone over a narrow range of concentration. So what we need to ask now is how robustly can the cells detect gradients? And so here's how we do that. We use a device, a microfluidic device. We basically build a little torture chamber to put yeast in, and we watch how they behave. So there's a top view here, and there's a cross section here. I'll talk about the top view here. So the screen here would be a microscope slide, and behind the microscope slide would be a cover slip, okay? And the cells are pasted on the cover slip, and we look from that side. What happens in this chamber is there are two inlets for fluid. This is a stream of yeast medium, which means sugar and other good stuff. which has no pheromone, no alpha factor. Here's another stream where it comes down in this direction, it does have pheromone. The dimensions of the chamber and the speed of the flow are such there's no turbulent mixing. It's not like waves or anything like that, or pouring one liquid into another. The flow is so smooth that the only way that pheromone gets from the top to the bottom as you go down this tube is it has to diffuse. And because diffusion obeys various physical laws, we know that the gradient will get flatter and flatter and flatter as you go down this tube. Okay? And so we can look at cells in specific positions. We know exactly what the pheromone gradient is, and we can monitor their behavior. If we take a cross section here, and we look at it down here, here's the chamber. The flow of liquid would be out of the screen towards you. Here are cells stuck to the cover slip that forms the roof of the chamber. It's about a millimeter wide. It's about 100 microns. That's a tenth of a millimeter deep. And here's our microscope lens that we do our observation with from the top. Okay? So that's the physical setup. And so what we're going to do now is we're going to monitor how accurately the cells detect gradients. So what I'm going to show you is, first of all, a graph that describes this. Then I'll show you a diagram. And then finally, I'll show you a movie. Okay? And before I show you the graph, I need to show you some pictures. And those describe the fact that global integration implies that there is a threshold for polarization, that above some critical concentration, cells can't polarize. And what that means is, remember the earlier diagram here, there's no net direction. We're going to deposit one particle on this side of the cell. Two things can now happen. If this particle falls off or diffuses within the plane of the plasma membrane, you can end up going backwards to this state here. And you can fluctuate, you can go back, you can go forth, you can go back. But if the asymmetry in the signaling particles, this side of the cell being higher than this side over here, gets to be strong enough, once you get here, you can never go backwards. And so after that, you've polarized and you're stuck. Okay? And so we can ask, can we detect cells crossing that threshold, and does a gradient, which imposes a net preference to end up on this side of the cell as opposed to this one, make it easier for them to cross the threshold? And so here are a set of pictures of very tiny cells. We've done something that is confusing. The outline of the cells in dotted line, we've inverted the contrast. So now it's the black that tells you where the components of the polarosome, in particular SPA2, are. These images are 20 minutes apart, and what you'll see as you walk across here, and the arrows will help guide your eyes, is that sometimes the black dot is in one part of the cell and sometimes it's another. It moves around. We're going to keep going in the movie. We're going to go all the way down, and we keep going. And here we are as we come back. And finally, at this time point here, the cell starts to polarize reliably in one direction. And now it's polarized. It shows polarized growth, and it makes it shmoo going in this direction. This is a uniform concentration of 1 nanomolar pheromone. We can compare that with this concentration over here, which is an average concentration of 1 nanomolar, but there's a gradient. The pheromone concentration is about 10% higher on this side of the cell than it is on this side over here. Okay? These are two cells, a mother and her daughter, and with the same sort of time points, we can see that much earlier, by about here, both of these cells have decided which direction to polarize in, and they're already reliably polarizing and shmooing. So having a gradient helps cells break symmetry, and it tells them which direction to do it in. And the question is, how reliably does that work as we change the pheromone concentration? So here's the summary cartoon. These are cells in the flow chamber, so the flow would be going along like this. And this is the lowest concentration of pheromone. This is the highest. There's a concentration of pheromone that is so low that the cells are simply oblivious to it. There's a higher concentration, which is the first thing you'll see in the movie, where the cells, instead of budding in their preordained directions, bud towards the source of pheromone. There's a concentration where the cells just grow symmetrically. They get bigger and bigger, like beach balls, without either budding or making shmoos. There's a narrow range of pheromone concentrations from 1 to about 5 nanomolar, where the cells shmoo up the gradient. And when the concentration gets high, like that movie I showed you, the cells get confused and shmoo in lots of different directions. So here comes the movie. Flow is going to be in this direction here. The high concentration of pheromone is here, the low is here. The movie's going to loop through. This is the time here in hours and minutes. This is distance, that's 10 microns. And what you're going to see is each time the white circle is going to draw your attention to a different set of cells. So I'll walk you through. Here are cells that are going to bud up the gradient. This cell makes a bud up the gradient. Here are the basketballs getting bigger and bigger. Here, for example, look at this cell as a cell that shmoos straight up the gradient. Here is a cell that starts in the wrong direction but turns up the gradient, and these are the hopelessly confused. Okay? And I'll just show you that again. Here we go, and we look at cells that bud up the gradient, the beach balls, the guys who are liable to find partners if there was a partner there instead of a chemical source of pheromone. Here are cells turning up the gradient, and here are the cells that are confused. Okay? And here's the graph that describes what happens. Here is the pheromone concentration. This is the logarithm of the pheromone concentration, so log 0 means this is 1 nanomolar pheromone right here. We're plotting the difference between the angle in which the cell's polarized, which is this direction here, and the direction of the pheromone gradient, which is straight up and straight down. We do the same thing we did before. We measure that angle theta for every cell. We convert it to a cosine. We average the cosines and we plot that. At concentrations below 1 nanomolar, most of the cells bud, which is this line going up here. At concentrations above, they shmoo, which is this line going down here. And what you will see is they do their best job at exactly 1 nanomolar pheromone, and the accuracy falls off very fast after that. So what we said as a prediction should be true is true, that the cells can't robustly detect pheromone gradients. There's a narrow range, maybe 3- or 4-fold here, where most of the cells go in roughly the right direction. Outside that range, if the pheromone concentration gets too high going in that direction, or it gets too low going in that direction, the cells can't polarize properly. And if that's true, how is it the case that cells can mate successfully under these very different conditions? And I'm going to try and explain to you why that's a problem. And this work depended on a collaboration with physicists in Israel, Noah Rappaport and Nama Barchai. And it comes from a very simple argument. So what we're going to do is we're going to imagine cells sitting on the surface of an agar plate. So they sit on top of a vast, relative to their own dimensions, thickness of agar. So molecules diffuse in what is effectively 3 dimensions. Here is an A cell, here's its pseudo or an alpha cell. There's a very faint pink wash which you may not be able to see, which describes the pheromone concentration falling off linearly as the distance away from the cell increases. Here is the same or a similar A cell surrounded by 6 alpha cells. And now the pheromone contribution from all the cells in the neighborhood adds up. And so the pheromone concentration that the A cell sees has gotten higher. Now we're going to add another ring of cells. The pheromone concentration has gone up some more. And we're going to add another. And as we keep doing that, the pheromone concentration goes up. The rate at which the pheromone diffuses is such that you could go way beyond the walls of this room and have the pheromone concentration keep going up. Not by increasing the density of these alpha cells, but just by increasing the geographical range over which they exist. Okay? And so this little bar here is meant to indicate schematically the concentration of pheromone. And there's a simple way of understanding this. So that is that if we think of a single cell, so let's take this cell right here, okay? We can ask how much pheromone does that cell contribute to the A cell that is right there? And the physics of diffusion are such that that contribution gets smaller and smaller as 1 over the distance, the radius if you like, between this cell here and the A cell there. But because of geometry, it's also true that as we keep building these concentric rings, going from this one to this one to this one, the number of contributing cells in each ring goes up as a function of r, because the circumference of a circle is pi times 2 pi times its radius. So we can ask what the contribution from each ring is by multiplying r by 1 over r. This is a sort of math I can do. That gives 1. And what that means is each ring contributes the same amount of alpha factor, so that if there are 2 rings instead of 1, the cells see twice as much alpha factor. And if there are 3 rings instead of 1, they see 3 times as much alpha factor. But I just showed you data that shows that cells don't do very well over a range of pheromone concentrations. So how is it that cells mate perfectly well like this and perfectly well like that? Okay? And this is where bar 1 comes back into the equation. Everything before this was done in the absence of bar 1 because we wanted to know what the pheromone concentration was. We didn't want the A cells to be eating the pheromone. But now we need to think about how things work. And so to do that, we want to know what pheromone concentration cells actually experience. Right? We can't build recording devices that are sufficiently small and sufficiently sensitive. So we ask the cells to tell us what concentration of pheromone they experience. And the way we do that is we take the promoter for a gene involved in mating. So we use this promoter here, which is called Fuz1. Fuz stands for fusion, so this is a protein involved in the cells fusing with each other. We fuse green fluorescent protein to the promoter of this gene, and so we can measure the activity of the promoter in arbitrary units as we change the pheromone concentration. Right? Lots of people have done this before us. And so as the pheromone concentration goes up, starting at 0 nanomolar pheromone, what we see happens to the level of the activity of this promoter is it just keeps going up, and eventually it saturates. We know from the experiments in the microfluidic chambers what range of concentrations is the one where cells detect gradients accurately. It's this narrow range here in this green wash here. And we can draw a line and we can say, okay, this is the highest level of activity that is compatible with detecting pheromone gradients. And right down here, it's right here, is the lowest, and we can draw a line across. If we see the activity of the promoter higher than this line or lower than this line here, the cells aren't going to be able to detect gradients. And the idea is that bar 1 helps the cell minimize the variation of the pheromone concentration at its surface as the local density of cells changes. And we're going to argue that the way that that happens is, here's an alpha cell. MF alpha 1 is the name of the gene, one of the genes that makes alpha factor. The cells make alpha factor. They bind to the A cell, so this black horseshoe is designed to depict the alpha factor receptor. And when they do that, they stimulate things inside the cell, that whole molecular signaling pathway I showed you before. We can ignore all its details. We just say, one of the things that these cells do is they induce the expression of bar 1, so these A cells will make their Pac-Man to chew up some of the alpha factor. Okay? This cell is being weakly stimulated. It doesn't make so much bar 1. Now, we imagine a cell that makes more alpha factor because this arrow has gotten thicker. It's stimulating the A cell more strongly. The A cell induces the expression of the bar 1 gene more strongly. You make more Pac-Mans and you help to minimize the extent to which the alpha factor concentration changed as this cell here opted to produce more and more alpha factor. So if you like, it's a way of blunting the change. It's sort of reducing the volume variation on your headphones. Okay? So if that's the right idea, there are a bunch of things that have to be true. The first thing has to be true that the expression of the bar 1 gene has to be regulated by pheromone. So this is the same sort of graph that you just saw. We have differences in pheromone concentration along this axis here. We have the activity of the promoter, which we measure by fusing the gene for a fluorescent protein to it here. This is the data I showed you before for the Fuz1 promoter. This is the bar 1 promoter, and what you can see is it is even more strongly regulated by pheromone than the Fuz1 promoter is, and in particular, it's induced strongly at much lower concentration of pheromone. So this is a sort of canary in the coal mine gene. Very tiny amounts of alpha factor will induce this gene very strongly, which is just what we need it to do in the sort of model I just showed you. Okay? And we can do an experiment where we ask what happens if we compare the activity of the Fuz1 promoter in 2 sorts of yeast cells. These cells here, again, this is data I've shown you before. These are cells that lack the bar 1 gene. These are guys that have the bar 1 gene. Okay? And what you will see is that at every concentration of pheromone, the cells that have the bar 1 gene induce the Fuz1 promoter less strongly. That means they see less alpha factor at their surface. And in particular, if we draw this gradient this line out that predicts where they will detect gradients. They ought to start detecting gradients at this pheromone concentration down here, and they still ought to be able to detect them all the way out here. So we can ask, is that true? Is this the gradient detection range for cells that can make bar 1? And so this is the same experiment I've shown you before. Here's our pheromone gradient, high to low. This arrow describes the direction of the gradient. This arrow here is the direction in which the cells polarize. We measure theta, the angle between them. We average the cosine of the angle, and we plot how well cells that either lack in white or have in black, bar 1, polarize in various different concentrations of pheromone. So this is 1 to 5 nanomolar, that range of pheromone concentrations. They do about the same. And now as the pheromone concentration goes up, as I showed you before, the white guys, the guys that lack bar 1, wimp out. The black guys who have bar 1 detect pheromone gradients over a much wider range of concentrations. And if you do experiments to ask, does that allow the cells to mate under a wide range of conditions, the answer is yes. Okay? And so what I hope I've done today is to tell you how scientists pick different models for how a process works. They discriminate between one model and another. They find problems, if you like, with that model, things that need more explanation. So in science, it's typically true that every answer produces more questions than you originally started out with, and how we go towards solving one of those questions, asking how it is that yeast can enjoy sexual activities with each other under a bewilderingly wide range of conditions. Thanks.